Hello, and welcome back to Introduction to Genetics and Evolution. We've been talking so far about mapping complex traits. We first talked about some of the principles associated with such mapping of complex traits, and we've also talked about how you do it using a cross. And we use this trajectory of associations. Now, let's look at an alternative approach. With simple genetic traits, we talked about mapping using data from overall populations. Well, we can do the same thing here in the case of complex traits. Now, you may be thinking first, why would we want to do this? Well, when you're QTL mapping in a crosser pedigree, what happens is you're localizing the genes affecting this difference between the original strains or people. So let's say you're using a pedigree, you're looking at, where are the genes contributing to breast cancer predisposition in this particular family. So let's say you're mapping a gene, and let's say in this case it's diabetes rather than breast cancer, in one family, and you map it to chromosome 18, arm P, location 22. Let's say there's a very strong effect next to a marker. Then you look in another family that has diabetes, and you see absolutely no effect to this region. Even though they have a lot of diabetes, and even though it is clearly running in the family, so it has a genetic component. Why would you see this dichotomy? Why would it have a strong effect in this family and no effect in this family? Well, the answer is, again, there are many different genes that can contribute to disease predispositions. And you may see in one family, there's a particular mutation that's contributing to diabetes predisposition, and there's a completely different one in another family. So when you study a trait in one single family, you're only identifying what's causing that variation within that family. It's important now to look at the broader population if you want to understand what causes diabetes more broadly. So the other approach that people use is this mapping in populations by association study. And a common variant of this is referred to as the genome-wide association study. This has become extremely popular lately with the ability to examine markers across the whole genome simultaneously, and of course with having a human genome sequence. Now, we discussed this in general in the context of simple traits, and the same principle applies as before. If a marker is close to the disease-causing gene, then individuals having one allele will be more likely to have the disease than individuals having the other allele. So there is an association between genotype at the markers and likelihood of disease, or whatever the phenotype is. The same principle is true not only in simple gene things, but also in multiple genes, such as disease. So what people do, especially with this genome-wide association study aspect, is they test many, many, many markers, right? Not just 3, 5, 10, 100, or even 1,000, but many, many markers. And what you'll find is that some will have an allele associated with the disease. Now, they're not going to have huge associations, kind of like some of the things we talked about before. You may see something like this. This would be a very large association in this case. That big A individuals have probability of getting the disease of 10%. Little a individuals have a probability of getting a disease of 1%. That actually is a very large effect relative to most of these sorts of things. Now, what happens is you do some sort of statistical test, such as maybe a chi-square, and this will say that this difference is unlikely to have happened by chance, given that you've sampled hundreds or thousands of individuals and you see this big an association. Okay? In contrast, many other markers will have no association. So this B marker, for example, may have a probability of getting disease of 1.5%. Little b has a probability of 1.6%. So the difference between big B, and little b is pretty trivial in terms of likelihood of getting the disease. Now, we discussed log plots when we were talking about genetic crosses. The same thing would apply with pedigrees. We do something very similar with association studies. We have these sorts of plots of association. But just like with simple traits, you can actually be much more fine scale because you're looking at the scale of the distance between these recombination hotspots, just as before. So this is an example. This is a mapping study from February 2012 and published in Nature Genetics of azoospermia in Chinese men. So these are people who are having sperm difficulties. And you see here this LOD score on the y-axis and position on the chromosome on the x-axis. And we see here there's a bunch of markers that have a strong association with this trait, whereas out here there are markers that don't have any association. So if we were going to do something along the lines of this A and B, these might be the big A individuals, right? Versus little a, where there's a strong association. This might be where B is, where there's basically no association. Okay? Now, what often happens, you find, since this is a complex trait, not one, but many possible factors. So this is from that same issue of Nature Genetics. This is just the title of the paper, A Genome-Wide Association Study in Han Chinese identifies multiple susceptibility loci for IgA nephropathy. And you see there's one on chromosome 8, one on chromosome 17, one on chromosome 22. Again, we're looking at the case of complex traits where there are many, many genes that have allelic variation or genotypes that are associated with the phenotype. Now, there's a problem that comes out with genome-wide association. This problem is the issue of multiple comparison. Now, those of you who are at Duke have seen this in lab, but let me tell you more generally, the chi-square test will give you a probability of getting an association by chance. I just mentioned that in the previous slide as well. Now, if you roll 2 dice, imagine you have some just standard 6-sided dice. You roll them and you get snake eyes twice in a row. Are they loaded? Well, you may think the answer is yes, because the probability of getting snake eyes twice in a row is very low. In fact, it's about 0.1% chance that you get snake eyes twice in a row. It's not 0, but it's very, very unlikely. So let's think of this in the context of looking at markers across the whole genome. How likely is it that you get something very improbable? Well, if you rolled the dice 1,000,000 times, what is the chance you get snake eyes twice in a row sometime over the course of that million? Well, the answer is it's about 99.99% chance that you should get that somewhere in there. This is a problem associated with genome-wide associations. That if you're testing a million SNPs, 1,000,000 of these markers for an association with a disease of interest, you're very likely to get an association that looks strong by chance when you do the statistical test, but there's no biological basis to it. That in fact this region has no effect whatsoever. There's no QTL there that's contributing to the trait of interest. But since you're testing so many times, the odds of you getting something that seems unlikely enough to be suggestive of a real effect is very high. So as a result, instead of using the standard 5% probability cutoff that people often use in statistics, people often use things like a 0.00001% probability as a cutoff. This is a way to work around this problem. So again, at that point, if you see an association, you have very high confidence that there's probably some sort of gene near there. The downside to this is that it requires a very strong association. Probably, and this probably happens a lot in genome-wide association studies, you actually see some associations, but you throw them out because you say not strong enough to be statistically convincing. So we end up losing a lot of good associations because we can't distinguish the good ones from the bad ones, except in these very strong cases. So let's contrast these two types of studies, pedigree cross versus population. Again, with a pedigree cross, you're finding genetic factors that cause differences in that family. So you have high power for a small number of genes because probably within a particular family, there's not tons of genes with allelic variation contributing to diabetes, breast cancer, nephropathy, whatever. In contrast, a population association study is trying to find genetic factors causing variation across the whole population. Now, typically here you have much lower power because you're looking at different causes throughout, but you can find potentially more genes, and if you do find them, you can localize them much more precisely, as I mentioned before. Now, you may be wondering, why is it that you have so much lower power? Aside from the statistical aspect, why do you have lower power? Well, there's another facet in here. In crosses and pedigrees, you're limited to mapping the genes with effects in that crosses. Okay? You're limited to mapping genes with effects in that cross. But the alleles you're mapping are initially abundant, right? If you're crossing, say, an individual with big Z, little z, across the little z, let's say this is the one that actually causes the mutation. In that initial cross, the fraction of individuals that have it is 25, or fraction of chromosomes, that have the mutation is 25%. So your ability to detect it is very high. Let's say in a population, though, you can theoretically map alleles at any gene causing this effect, and there's probably a lot of them doing it. But you may have many different very rare variants causing the same effect or disease. So in this case, let's say, for example, this capital A and this capital Z both cause the disease. They're each initially very rare, so it's very hard to see their effect. It's because of this rarity. Now, genome-wide association studies work well when you're mapping common disease variants. But an open question is, are most disease variants common? In fact, several people have argued that, in fact, most disease-causing variants are quite rare. And there's often different rare mutations that cause the same disease. And I refer you in this context to an interview that I have online with Professor David Goldstein from Duke University about this idea, about rare variants causing disease. There's another confounding factor that comes along with both of these kinds of studies, and that is you can see differences among ethnic groups for association. That sometimes a marker allele is associated with a disease in one ethnic group, but it's not associated when you look at another ethnic. That if you map something in the Han Chinese, you may see an effect associated with this particular region. In contrast, when you look at, say, a Latin American population, you may not see that association. Let me give you a real example of this. This is from type 2 diabetes. And in this case, the initial study that was done using East Asian subjects. And what they found is that there were 4 SNPs that were identified that have a pretty strong association with this diabetes in this set of East Asian subjects. In contrast, when they looked in European populations, there was one particular SNP, one on chromosome 11, that had no detectable effect in Europeans. So clearly there was some sort of difference between these two groups. There are many possible explanations for this. One of them is, we mentioned before, epistasis. There's interactions between genes. That, for example, another variant may be very common in Europeans that makes it so this particular gene variant that's near the SNP that's causing diabetes in East Asians doesn't have that effect in Europeans. That's one possibility. Another possibility is the allele at that gene which is causing diabetes in East Asians just isn't found in European populations or is very rare. There's a lot of possible explanations out there. But importantly, we can't immediately extrapolate and say, oh, there's a gene that's causing this disease, and just assume that that's causing it across different families, across different ethnic groups, across different continents. So mapping is often a very complex task, but it's obviously very worthwhile because we have made inroads in identifying many genes that are associated with disease, some of them with fairly large effects, though many with much more moderate effects. These are the genes that are actually used in those studies that you get from companies like 23andMe that takes samples of your saliva and tell you your disease predisposition. They're using this sort of data to tell you what your likelihood is of getting the disease. Thank you very much. I hope you enjoyed this video.